Hello everyone, this is a presentation on the role of DNA biomechanics in regulation of gene expression by Anushree, Sahana and Sangeeta. Search terms, when we came up with regulation of gene expression in DNA, it produced a lot of results, as you can see. I chose this article because I understood the title, and the abstract displays how DNA is affected via many chemical pathways, and it showed terms I'm familiar with. As you can see, this is the copy of the abstract from the article. The keywords here are mechanical features, biomechanics, DNA mechanics, mechanoregulation, mechanome, and mechanotransduction. Introduction. Mechanosensitive ion channel, protein kinase, small and large G protein are all mechanotransduction pathways in which transfer mechanical potential on plasma membrane to chemical signals that initiate cellular response, producing force-dependent changes in gene expression further subsequential of signal transduction. Localized protein binding and aggregation are when proteins bind to the DNA and how they come together as a whole from various sources. Tension dependent means how the force is applied to the DNA and behaves when the protein molecule that binds is far and helps in functioning as a whole. Mechanical effects and the chromosome involves how the tension is applied on the DNA or the nucleus can be seen as influencing chromosomal structure. The outlook suggests on modernization techniques for exploring DNA biomechanics. Local protein binding and aggregation. This is a figure that displays, part A displays proteins that are bound to DNA can produce a twist and tension along the molecule. One molecule can be increased rate of aggregation driven by a need to lessen the deformational cost in energy associated with their binding. Part B shows certain restriction endonucleases like EcoRI in yellow must first bend 50 degrees in the DNA before it can cut and display an activity level dependent upon tension in the molecule. Others like BAMHI in green can cause slight distortion to DNA and show little sensitivity to tension. Part C shows disassembly of nucleoprotein filaments is a vital step in recombination and is strongly tension dependent. The filaments are steady as long as the RAD51 proteins are bound to the ATP in green. The RAD51 monomers can hydrolyze ATP in purple. They dissociate from the filament once a final monomer hydrolyzes ATP. Dissociation continues up until the point at which the next RAD51 monomer is found bound to ATP.
Tension-dependent control of distant cooperator process. DNA looping used for combining proteins from promoter to initiation for transcription. To enhance this in eukaryotes, supercoiling is used. This is driven by thermal fluctuations and intracellular actions that contingently bend and twist DNA. Two binding sites come close to form the loop. Thermal fluctuation force need 0.1 picon-newton on consistent length to work, or female-Newton force exerted. It stops thermal fluctuation, prevents loop formation, transcription levels. The C figure shows type 2 restriction endonucleases like SAU3AI cut when distant restriction sites encounter an inactive enzyme and exhibit a strong suppression of activity at tensions of below a picon-newton. Protects unwanted division of gene if methylated. Mechanical effects and chromosome. Tension plays a corresponding role as it directs binding of nucleosomes to DNA, hence crucial detriment of nucleosomal positioning. Mechanical stress affects nuclear DNA. Temporal mechanical stress formed by molecular motors can propagate through chromatin fiber and cause local changes to DNA. During the transcriptional activity of RNA polymerase, additional TOK is generated from the processive motion of the polymerase. This TOK results in positive supercoils along the template DNA downstream of the polymerase that may help unwind nucleosomes, clearing the DNA for transcription. Similarly, negative supercoiling upstream of the polymerase might aid in the reassembly of nucleosomes with the template DNA.
Mechanical effects and chromosome. Structure architect in the cell control gene expression, which makes certain regions accessible to direct expression inhibition in group of genes. Thus, chromosomes are involved due to its own packaging and the dependent on mechanical properties of the chromatin. Chromosomes with the nucleus is packed tightly, so in the internal driven mechanical forces cells propaganda of the chromosomes. In eukaryotic chromosomes, fibers cause histone modification eliminates or loss of the non-histone structures. Outlook for exploring mechanics of genes expression. Modern techniques involve inserting microneedles into living cells for excretion of mechanical force directly. Image B, optical tweezer, look spexity apply and force due to difference in refraction in index trap lipid dropped, active transport. In cell, high laser heat produced cause photo damage to cell. Image C, magnetic tweezer, high spexity by using functionalized supermagnetic particles in cell requires 3D control. Microfabricated magnetic tweezer, 2D, control to manipulate micro-injected supermagnetic beads inside of the HeLa cells, measure elasticity and viscosity. Requires 500 nanometer beads to use while nanoprobe didn't move internally and can disturb nucleus function, but can bind to histone to apply mechanical force on chromosome and the C coordinate affects to gene expression.
In conclusion, more effort and technological advancements are being made to explore mechanical tension to be determined directly and specifically in living cells in order to understand and lay the foundation for a new exemplar of how we connect with and explore the cellular world.